You have probably heard this statistic 1,000 times,
Mhm.
It's in textbooks, it's in articles, it's practically pop science gospel at this
Wait,
point.
absolutely.
The human brain has 100 billion neurons. It's this nice, round, staggering number, but there's this tiny problem with it. It's
Wow.
completely wrong.
Yeah, completely wrong. I mean, it really is a testament to how easily a nice round number can just become unquestioned dogma.
Right. Welcome to the deep dive. Today, we're going to tear down everything you thought you knew about the brain. And we're starting with how a bitter rivalry over a microscope slide unlocked the actual 86 billion biological processors inside your head.
For decades, that 100 billion figure survived without like a single solid empirical source backing it up. It was just assumed.
Assumed until 2009, right? When the Brazilian neuroscientist, Susana Herculano Husel, finally decided to do a proper head count.
Right.
I was reading about her methodology, and she used something called the isotropic fractionator, which, I mean, it sounds incredible, almost like a sci-fi ray gun.
It does sound like sci-fi.
But I am struggling to picture what it actually does. Like, how do you count cells in something as incredibly complex as a human brain?
Well, the main problem with the brain is that it's highly irregular. You can't just count the cells in a tiny slice and multiply it by the volume of the whole brain.
Because it's not uniform.
Exactly. Some regions, like the cerebellum, are densely packed with neurons, while other regions are much more sparse. So Herculano House's genius was realizing that to get an accurate count, she had to destroy the brain's architecture entirely.
We destroy it.
Yeah. The isotropic fractionator essentially dissolves the brain tissue. It breaks down all the cell membranes, but it leaves the cell nuclei completely intact.
So she basically turned a solid human brain into a giant beaker of uniform liquid soup.
Precisely. A brain soup. By making it a perfectly mixed, uniform suspension, she could take just a tiny drop of that soup, count the neuronal nuclei versus the non-neuronal ones, and then accurately scale that number up to the total volume of the original fluid.
That is so clever. And the real number she came up with was roughly 86 billion neurons.
Right.
Which, I mean, that might sound like we just misplaced a few billion as a rounding error, but 14 billion neurons is basically the entire brain of a baboon.
That's a massive difference.
It really is. And she also debunked this other huge myth, right? The one saying you have support cells, glial cells, outnumbering your neurons 10 to 1.
Oh yeah, the 10-to-1 myth. We now know it's actually about a 1-to-1 ratio, depending on the specific brain region. That fundamentally changes our entire understanding of the brain's composition.
So it's not mostly filler.
Exactly. We are not carrying around a massive excess of purely supportive tissue. Your brain is actually a highly efficient, densely packed network.
Okay, let's unpack this. To understand those 86 billion individual neurons sitting inside your skull right now, we first have to understand how scientists figured out the brain wasn't just one giant fused blob of tissue in the first place.
It's a wild history.
Because looking back at the sources, people used to think the nervous system was just a single, continuous organism.
Yeah. For most of the 19th century, the dominant view was called the reticular theory. Scientists looked at nervous tissue and they just saw a continuous fused web of protoplasm. They called it a syncytium.
A syncytium.
Right. Under that view, the very concept of a neuron as a single bounded individual cell didn't even exist.
But how did we ever break away from that? Because,
Huh.
if you look at brain tissue under a normal microscope, it just looks like a dense, tangled mess of spaghetti. You can't see individual cells.
You really can't. And the breakthrough is, honestly, one of the greatest detective stories in biology. It started in 1873 with an Italian physician named Camillo Golgi.
Oh, I recognize that name.
You probably do. He was experimenting in his kitchen laboratory, of all places, and he invented this silver chromate staining technique. He called it la reazione nera, the black reaction.
The black reaction. Sounds ominous.
Well, the brilliance of this stain is its incredible inefficiency.
Inefficiency.
Yeah. By pure chance, this chemical process only stains about 1 to 2% of the cells in a sample.
Oh, wow. But it turns those specific cells entirely black against a completely transparent background.
Exactly. So instead of a dense forest where you can't see the trees, you get a silhouette of just 1 or 2 perfect solitary trees. You can see every branch.
That's amazing.
But the irony here is that Golgi used his own revolutionary technique to argue in favor of the reticular theory.
Wait, really?
Yeah. He looked at these sprawling, deeply stained structures and concluded they must all be fused together at their ends into a continuous net.
Enter the Spanish neuroanatomist, Santiago Ramon y Cajal.
Yes, Cajal.
He takes Golgi's silver stain, and he just goes to work obsessively. Like he's meticulously illustrating brain tissue from birds, from embryos, from basically everywhere he can find.
He was a brilliant artist, too.
Yeah, and through this incredibly detailed work in the 1880s and 90s, Cajal builds the case that these are discrete individual cells with clear boundaries. They communicate by contact, not by being fused together.
Right. This became known as the neuron doctrine. And then in 1891, a German anatomist named Heinrich Wilhelm Waldeyer synthesized Cajal's massive body of evidence and officially coined the term neuron.
So was it like looking at a map where Golgi thought the nervous system was one giant continuous highway system, but Cajal realized it was actually millions of separate driveways with tiny gaps between them?
That is a perfect way to visualize the debate, and the tension over those metaphorical driveways really climaxed in 1906.
The Nobel Prize.
Exactly. The Nobel Committee decided to award the prize in physiology or medicine jointly to both Golgi and Cajal.
Oh, that must have been awkward.
Incredibly, you had two men who fundamentally disagreed on the basic unit of the nervous system, forced to share a stage. Golgi actually used his Nobel lecture to attack Cajal's theory.
Wow. Right there at the ceremony.
Right there, even though the scientific consensus was already tilting decisively toward Cajal.
Well, Cajal's discrete cells won out. We know today that you have 86 billion separate neurons. But, science rarely wraps up that cleanly.
It doesn't. What's fascinating here is that Golgi was actually partially right.
Really? How so?
Much later in the 20th century, using electron microscopes, we discovered electrical synapses. They're also known as gap junctions.
Gap junctions.
Yeah. In these very specific, specialized connections, adjacent neurons actually do share direct electrical continuity. Ions flow straight from the cytoplasm of one cell into the next.
So they are physically connected.
In those specific spots, yes.
Yeah.
It was a partial, limited vindication for the reticularists that nobody in the 1890s could have possibly anticipated.
That is wild. Okay, so we have these millions of separate driveways, right?
Right.
But how does a single microscopic, basically a salty water balloon of a biological cell, actually move a thought from one side of itself to the other?
Well, the neuron solves that engineering problem by dividing its labor across a specialized physical architecture.
It's split into parts.
Right. 4 main zones. A thought begins at the receiving end, the dendrites. These are the branching, tree-like extensions coming off the main body of the cell. Their entire job is to receive incoming signals from other neurons.
And some of these dendritic trees are massive, right? Like I was looking at the cerebellar Purkinje cell.
Oh, the Purkinje cell. First described by Jan Evangelista Purkinje in 1837, it is a marvel to look at.
It really is. It has this extravagant, flat, fan-shaped branching structure.
And a single one of those cells can receive well over 100,000 individual synaptic connections.
100,000.
Yeah. It perfectly illustrates that neurons are not just simple relay wires. They are actively performing massive computations. They're summing and weighing thousands of simultaneous inputs at any given moment.
So the dendrites catch all these incoming signals, but they need somewhere to process them, obviously. That funnel is the main cell body, the soma.
Correct. The soma contains the nucleus and the standard cellular machinery. This includes the mitochondria for energy and, interestingly, the Golgi apparatus.
Named after Golgi.
Discovered by our very same Camillo Golgi in 1898 while he was studying those exact Purkinje cells.
Of course he did.
So the soma tallies all the electrical inputs arriving from the dendrites. Now, a resting neuron sits at an electrical potential of about negative 70 millivolts.
Okay, so it has a negative charge at rest. And if the combined input from all those dendrites pushes the voltage up to a specific threshold, say around negative 55 millivolts, the cell decides to fire.
Exactly.
What actually happens at that exact moment?
The signal hits a specialized trigger zone called the axon hillock, and then the axon takes over.
The third zone.
Right. The axon is the transmission cable. It carries the electrical signal away from the cell body toward the next cell. And the scale of these axons is where the biology becomes almost unbelievable.
Unbelievable. How?
Well, some interneurons in your brain have axons that are a tiny fraction of a millimeter long. But consider your sciatic nerve.
Down the leg.
Yeah, you have single motor neurons with their cell bodies sitting in your lower spinal cord, and their axons stretch all the way down your leg to the muscles in your foot.
So in a tall adult, that is a single continuous cell spanning almost a meter.
A single cell, yes.
Which raises an immediate red flag for me. If I stub my toe, I feel it instantly. But if I have a microscopic, electrically leaky biological cable running from my spine all the way down to my foot, how does that signal not just fizzle out along the way?
That's the perfect question. The body evolved an ingenious solution. It's a fatty insulating sheath called myelin.
Myelin,
Yeah.
like insulation on a wire.
Exactly. In your peripheral nervous system, like your leg, this myelin is produced by specialized support cells called Schwann cells.
Okay.
In your central nervous system, it's produced by oligodendrocytes. They tightly wrap the axon to prevent the electrical signal from leaking into the surrounding tissue. But the real genius is that the myelin is not a continuous tube.
Right, it has tiny breaks in the insulation.
Yes, it's interrupted at regular intervals by exposed patches of the raw axon membrane. These gaps are called the nodes of Ranvier.
Nodes of Ranvier.
Named after the French histologist Louis-Antoine Ranvier, who described them back in the 1870s. Because of these specific gaps, the electrical impulse doesn't have to travel slowly and continuously through the whole cable.
It's like the difference between driving on a local back road, where you'd stop at every single traffic light, versus getting on the express highway and just flying between the major on-ramps.
Exactly that mechanism.
Yeah.
It's called saltatory conduction, from the Latin word saltere, meaning to leap.
So it leaps.
The electrical signal literally jumps from node to node, and the speed difference is staggering. I mean, an unmyelinated fiber conducts a signal at roughly 0.5 to 2 meters per second.
Pretty close.
Right. But a large myelinated fiber using this jumping method can conduct at over 100 meters per second.
Wow.
That high-speed highway is why you feel the stubbed toe almost instantaneously.
Okay, so the signal races down the axon at 100 meters per second, and then it hits the end of the line, the fourth zone, the axon terminal.
This is where the electrical signal hits a hard stop. We've gone from the receiving dendrites, through the integrating soma, down the transmitting axon, and now we reach the output.
Right.
But before we cross the gap to the next cell, we need to understand what that electrical signal actually is.
Good point, because we keep talking about an electrical signal, but, there are no copper wires in my arm. What is the actual spark?
It is called the action potential, and it relies on a very specific type of protein built right into the cell membrane called a voltage-gated ion channel.
Voltage-gated.
Right. When the voltage hits that negative 55 millivolt threshold we mentioned earlier, it triggers sodium channels to open. Positively charged sodium ions from outside the cell just rush in.
Because they want to balance the charge.
Exactly. And that wildly spikes the membrane voltage all the way up to roughly plus 30 millivolts.
Okay, I am going to admit some confusion here. How does a biological protein channel just know when to open? I mean, it doesn't have a brain of its own.
No, it doesn't. It just comes down to basic physics. The proteins that make up these channels are themselves electrically charged.
Oh, I see.
So when the electrical field across the membrane changes, the physical shape of the protein is literally forced to bend and contort. The voltage change physically rigs the channel open.
That is fascinating.
So sodium rushes in. Then, just a fraction of a millisecond later, voltage-gated potassium channels bend open. That lets positively charged potassium ions rush out of the cell, which drops the voltage right back down.
But now the ions are all in the wrong place. The sodium is trapped inside, the potassium is trapped outside.
Which is where the heavy lifting comes in. The neuron deploys the sodium-potassium pump.
The pump.
Yeah, this is a molecular machine that constantly burns cellular energy, ATP, to forcefully exchange 3 sodium ions back out of the cell for every 2 potassium ions it brings back in.
It's manually resetting the trap.
Exactly. It painstakingly resets the ionic balance so the neuron can fire all over again.
How on earth did we figure out the molecular mechanics of this in the mid-20th century without modern computers? I know 2 British scientists, Alan Hodgkin and Andrew Huxley, published the mathematical model for this in 1952, but how did they measure electricity inside a microscopic cell?
Well, they couldn't use mammalian cells. They were way too small. Hodgkin and Huxley had to use the Atlantic squid.
A squid?
Yeah. The squid has a giant axon that controls its water propulsion jet. And this axon is nearly 1 millimeter in diameter.
1 millimeter. You could practically see that with the naked eye.
You can. It was the only nerve fiber in the animal kingdom large enough that they could physically insert tiny measuring electrodes straight down the middle of it.
That's incredible.
And that physical measurement earned them the 1963 Nobel Prize.
Here's where it gets really interesting. If you think about everything you just explained, a neuron is almost exactly like a rechargeable battery.
It really is.
It discharges this massive spark of electricity by letting the ions flow, and then that sodium-potassium pump has to frantically recharge it, forcing the ions back against the gradient. before it can fire again.
The internal mechanics are very much a rigid, reliable electrical battery, but that electrical spark eventually hits the axon terminal, and it cannot jump across the empty physical space to the next cell's dendrite.
So how does the signal get across the driveway?
It converts from an electrical signal into a chemical one.
Ah,
The tiny gap between the cells, the synapse, a term coined by British physiologist Charles Sherrington in 1897, that gap is bridged by neurotransmitters.
okay, so it switches to chemicals.
Yes. When the action potential hits the end of the axon, it forces tiny sacs called synaptic vesicles to dump these chemical messengers out into the fluid gap. They float across and bind to receptors on the next cell.
We proved this chemical jump with Otto Loewy's frog heart experiment in 1921, didn't we?
Yes. It is one of the most elegant experiments in all of biology.
Oh, walk me through it.
So, Loewy took a beating frog heart, complete with its vagus nerve still attached, and placed it in a bath of physiological fluid. He electrically stimulated the nerve, which slowed the heart rate down.
Makes sense.
Then he simply took the fluid bathing that first heart and applied it to a second, entirely separate, isolated frog heart.
With absolutely no electrical stimulation to the second heart.
None. Zero. And yet the second heart also slowed down.
Wow.
Loewy proved definitively that the electrical signal in the first nerve had released a physical chemical into the fluid. He called it vagustov, which we now know as the neurotransmitter acetylcholine.
And the legend is that the design for this experiment reportedly came to him in a dream, right?
Yep. He woke up, scribbled it down in the middle of the night, and ran to the lab the next morning to test it.
That is just so cool. So while the inside of the neuron is this rigid electrical battery, the outside communication is this incredibly flexible chemical soup. We're talking about neurotransmitters like dopamine, serotonin, glutamate. And because it is a chemical soup, it means the connections can be modified.
Exactly. This completely upends the misconception that neurons are just passive wires in a fixed, static circuit.
Right.
The reality is synaptic plasticity. Based on how often that chemical synapse is used, the connection physically strengthens or weakens.
So it adapts.
That dynamic changing of connections at the chemical gap is the literal physical foundation of all human learning and memory.
Which also beautifully kills that tired pop culture myth that we only use 10% of our brain.
Oh, completely. Modern brain imaging shows widespread metabolic activity distributed across virtually the entire brain throughout the day.
Even when you're just resting.
Even then. Because even tissue that isn't actively firing a massive action potential is constantly maintaining that resting battery voltage. We know that if you injure almost any small part of the brain, you see a measurable cognitive or physical deficit.
Speaking of damage, I want to go back to the stubbed toe, or say a cut finger. If I slice my finger chopping vegetables, I might lose feeling for a while, but the nerve eventually heals.
It does. Peripheral nerves have an incredible capacity for regeneration.
How they do it.
They use a process called Wallerian degeneration. The Schwann cells we talked about earlier, the ones making myelin,
Right.
they actually help clear out the damaged axon debris and form a physical tube to guide the regenerating nerve fiber back to its target.
Wait, if my finger can heal itself and reconnect the wiring, why is a spinal cord injury permanent?
It is one of the most tragic biological trade-offs in the human body. Central nervous system axons, the ones in your brain and spinal cord, largely cannot regenerate.
Why not?
And it isn't because they forget how to grow, it's because the surrounding central nervous system tissue actively suppresses their regrowth.
Actively suppresses it.
Yes.
Well,
Researchers have identified specific inhibitory proteins. They're literally named things like no-go-loc that block axon regeneration.
no Kogo. It's a bit on the nose.
It is. The central nervous system prioritizes stabilizing its complex circuitry over repairing damage, which is why spinal cord injuries are typically permanent.
The brain is actively pumping out proteins to stop its own healing. That is devastating. But what about creating totally new neurons from scratch?
Ah, that is the topic of adult neurogenesis. For decades following Cajal's original doctrine, the medical consensus was that you are issued your full supply of neurons at birth and you can only lose them.
Right, that's what I was always taught.
But from the 1960s to the 1990s, evidence emerged showing that rodents and some primates could grow new neurons in specific areas, like the hippocampus, which handles memory.
Oh, wow.
But proving it in adult humans has been remarkably difficult.
Why is it so hard to just look at a human brain and see if new cells are forming?
Because you can't easily study living human brain tissue, you have to rely on post-mortem studies. And using different methods has yielded really chaotic results.
With what?
Well, some labs used atmospheric radiocarbon dating of the DNA left over from Cold War nuclear testing to see if the cells were born recently.
That is insane.
Other labs used marker protein staining. And even deep into the 2010s, major papers have come out completely contradicting each other on whether adults generate new neurons.
The debate continues. But when we lose them, the results are undeniable.
Absolutely.
Alzheimer's disease is the progressive death of neurons, especially in those memory centers.
Right.
And Parkinson's disease is the death of a very specific population of dopamine-producing neurons in a tiny midbrain region called the substantia nigra, which directly causes the tremors.
Yes. And multiple sclerosis directly attacks the architecture we just explored.
Or myelin.
Right, exactly. In MS, the patient's own immune system attacks and strips away the myelin sheath. Without that insulation, you lose the high-speed saltatory conduction.
The leaping.
Right. The electrical signals slow down or leak out entirely, causing weakness and vision loss.
So why can't we just fix the physical damage with medication?
Well, if we connect this to the bigger picture, it explains the entire modern paradigm of mental health treatment.
How so?
Most psychiatric drugs, antidepressants, antipsychotics, anti-anxiety medications, they do not target the structural cables or try to regrow myelin.
Because it's too hard to rebuild the architecture.
Exactly. It's incredibly difficult to rewire the physical architecture of the brain. But we can safely intervene in that microscopic fluid gap.
The chemical soup.
Right. They exclusively target the chemical synapse. We can block serotonin from being absorbed too quickly, or mimic dopamine, utilizing the brain's natural chemical flexibility to treat disease.
So what does this all mean? We started by throwing out the myth of 100 billion neurons, literally dissolving brains into soup, just to get a proper count.
Yep, 86 billion.
We traveled back to Golgi and Cadell, arguing over a stained microscope slide about whether the nervous system was a giant highway or millions of individual driveways.
And it turned out to be driveways mostly.
Right. We saw the beautiful branching dendrites catching signals, the soma tallying the math, and the massive electrical spikes racing down insulated myelin highways to squirt just a tiny fraction of chemical soup onto the next cell.
It's an incredible journey.
It is. The 86 billion tiny islands in your head are not just simple wires. Every single one of them is a dynamic biological processor constantly adapting and changing. That incredibly complex, fragile chain reaction is what generates your entire reality. I mean, it is exactly how you are understanding the sound of my voice right now.
It is a profound realization. And this raises an important question, an incredible modern application of everything we have discussed that we should probably leave you to think about. Remember Hodgkin and Huxley mapping the precise math of the electrical action potential in the giant squid axon?
Right, the 1963 Nobel Prize.
Well, because they cracked the mathematical code of how a neuron fires, modern science can now interface with it directly.
Like plug into it.
Yes. Brain-computer interfaces are no longer science fiction. We are currently putting microelectrode arrays directly into the motor cortex of paralyzed humans.
Wow.
By reading the electrical action potentials from specific populations of neurons, we can translate that biological math into digital code. This allows patients to control computer cursors or robotic limbs purely with their thoughts.
Wait. The same electrical spark that moves an arm is just running a machine.
Exactly. So here's the thought to ponder. If a single neuron's electrical output evolved to control a biological body, but we can now plug that exact same electrical output directly into a machine, where does the human nervous system end and the outside world begin?
Wow. We will let you sit with that one. Thank you for joining us on this deep dive into your own head. Keep those 86 billion neurons curious, and we will catch you next time.